Discover how microfluidic droplets revolutionize synthetic biology by creating millions of isolated reaction vessels for programmable biochemical production. Droplet-based synthetic biology uses microfluidic technology to create millions of tiny isolated compartments where biochemical reactions occur with unprecedented precision and control. Traditional synthetic biology relies on living cells, which introduce significant constraints and inefficiencies. Living cells must grow, divide, and maintain complex internal regulation systems that often interfere with engineered synthetic pathways. Cells actively suppress foreign genetic material, degrade engineered proteins through quality control mechanisms, and divert resources to native metabolic processes. These biological constraints result in slow growth rates, highly variable gene expression levels, and unpredictable cellular responses that severely limit production efficiency. Multiple cellular constraints stack up: division times measured in hours, metabolic burden from engineered pathways, complex gene regulation networks, and constant protein degradation. Microfluidic platforms generate millions of uniform droplets per second, each serving as an isolated reaction vessel. In microfluidic chips, aqueous reagent streams meet oil phases at precise junction points, where surface tension forces spontaneously form uniform droplets at kilohertz frequencies. Each droplet ranges from picoliter to nanoliter volumes, creating reaction chambers billions of times smaller than traditional test tubes. The technology produces thousands to millions of droplets per second, each with identical size, shape, and chemical composition, enabling massive parallelization. Each droplet contains purified transcription translation machinery that performs synthetic biology without living cells. Inside each droplet, purified DNA templates, RNA polymerase, ribosomes, amino acids, and energy molecules work together to transcribe genes and translate proteins without cellular interference. The central dogma proceeds rapidly. DNA is transcribed into messenger RNA, which is immediately translated into functional proteins by cell-free machinery. Without cell membranes, metabolic networks, or native gene regulation, the synthetic pathways operate at maximum speed with minimal interference. Droplet isolation provides multiple critical advantages over both cellular and bulk cell-free systems. Each droplet serves as an identical, isolated microreactor with precisely defined volume, identical reagent concentrations, and complete protection from cross-contamination. The uniformity of droplet volumes ensures that millions of parallel reactions proceed under identical conditions, eliminating volume-related variability. Physical isolation prevents molecular crosstalk between droplets while maintaining the crucial link between genetic templates and their protein products within each compartment. Droplet systems enable ultra-high throughput screening, testing millions of variants simultaneously with real-time fluorescence monitoring of each individual reaction. Droplets can host sophisticated genetic circuits that operate as autonomous biochemical processors. RNA-based logic circuits, riboswitches, and feedback networks operate autonomously inside droplets, creating tiny programmable biochemical processors with complex input-output relationships. Engineered riboswitches sense molecular inputs, RNA circuits perform logical operations, and feedback loops self-regulate, all executing autonomously within the droplet environment. Fluorescent reporters enable real-time monitoring of circuit outputs, allowing researchers to track gene expression dynamics and circuit behavior millisecond by millisecond. Microfluidic control allows systematic optimization of reaction conditions, droplet by droplet. Researchers can vary pH, ionic strength, temperature, and cofactor concentrations droplet by droplet, creating comprehensive parameter spaces for reaction optimization. By systematically varying conditions across droplet populations, researchers rapidly identify optimal parameters for enzyme activity, protein folding, or circuit performance. The total optimization space grows as the product of all tunable parameters, enabling comprehensive exploration of multidimensional reaction landscapes. Advanced microfluidic operations enable complex multi-step reactions and pathway assembly. Droplets can fuse on-chip, combining their contents to initiate sequential reactions, add substrates, or mix enzyme cascades in controlled temporal sequences. Droplet splitting enables reaction dilution, aliquoting for parallel analysis, or branching pathways that explore multiple synthetic routes simultaneously. By programming fusion and splitting sequences, researchers construct multi-enzyme cascades that mimic the complexity of cellular metabolic pathways without cellular constraints. Droplet systems dramatically accelerate protein engineering and directed evolution campaigns. Millions of enzyme variants are tested in parallel, each in its own droplet, with fluorescence-activated sorting rapidly isolating the most active or selective variants. Fluorescent substrates or biosensors report enzyme activity in real time, allowing microfluidic sorters to isolate top-performing variants from vast libraries at thousands of droplets per second. Droplet platforms routinely screen over 1 million variants per day, accelerating directed evolution campaigns from months to days. Droplet-based synthetic biology offers transformative advantages over both cellular and bulk approaches. Droplet systems combine unprecedented speed, precise control, massive throughput, complete isolation, fine-tuned optimization, and full automation into a single integrated platform. Reactions proceed 10 to 100 times faster than cellular expression, with control precision impossible in bulk systems while maintaining massive parallelization. By completely decoupling synthetic biology from living cells, droplet systems eliminate growth constraints, metabolic burden, and regulatory interference. Droplet-based synthetic biology represents a paradigm shift in how we design and execute biochemical production. Microfluidic platforms generate millions of uniform, isolated picoliter-scale droplets that serve as programmable biochemical microreactors. Purified cell-free machinery inside each droplet executes synthetic biology programs without cellular membranes, metabolism, or regulation. The combination of precise condition control, massive parallelization, and real-time fluorescence monitoring dramatically accelerates pathway optimization and protein engineering. Advanced droplet operations like fusion and splitting enable complex multi-step synthetic pathways and ultra-high throughput directed evolution campaigns. Droplet-based synthetic biology transforms biochemical manufacturing from an art into an engineering discipline with programmable, automated, and highly scalable production systems.